Good evening and warm welcome to our next session of human embryology, in which in the chapter 6 we shall be discussing about the placenta. Then followed by fetal membranes, then followed by the twinning. All right, we discuss placenta first. Fetal membranes. Next, twinning. That is the physiology of twin pregnancy. All right. So, as a matter of fact, as our habit goes, before discussing any class, we'll summarize what we are discussing today. I'll be discussing first about the implantation. So what is implantation? The growing embryo, the growing zygote, then morula stage I discussed, then blastocyst. So at the stage of blastocyst, implantation occurs, and implantation is nothing but it is the attachment of the embryo, the growing blastocyst or embryo, to the uterine endometrium, to the uterus. So imagine this is the uterus. So the growing embryo comes and attaches to the endometrium, innermost layer of the uterus. So this process you are calling it as implantation. We'll discuss what are the types of implantation. Interstitial implantation, where the human embryo undergoes it gets deep buried into the endometrium. Then we'll talk about an important structure or an important change that is occurring in the endometrium. Once the implantation occurs, the endometrium is called as decidua. You should remember this name, decidua. What is decidua? After implantation, the endometrium becomes decidua. After implantation, endometrium changes into decidua. This we will discuss. Endometrium has got a new name, decidua. Different types of parts of decidua. We'll discuss the different parts of and types of decidua. Then we'll discuss at this point of attachment of the embryo, there is a specialized structure that is formed partly by the mother and partly by the fetus, partly by the embryo and partly by the endometrium. And that structure you are calling, it is a plate-like structure, disc-like structure. You are calling it as a placenta. So we'll discuss details about the placenta. We'll discuss how the placenta is formed. What are the physiological aspects of the placenta, wherein the placenta is responsible for transport of nutrients, especially the oxygen and various other nutrients, the glucose, which helps in a nutrition to the fetus, growth of the fetus, and also removal of waste products. So my audio and video are clear? Yes. Next we'll discuss the parts of the placenta. The fetal tissue that takes parts in the formation of the placenta, we are calling it as a chorion. So one part of placenta you are calling it as chorion. What is chorion? Chorion is that part of the placenta which is formed by the fetus, which is contributed by the fetus, fetal tissues, fetal cells, fetus cells. Next we'll talk about the maternal component. The maternal component that is responsible for formation of the placenta. And here we have got trophoblasts. So the parts of the placenta, in that we'll discuss what is trophoblast. So we'll also discuss the two types of trophoblasts, two types of cells which contribute to the trophoblast, the cytotrophoblast and the syncytiotrophoblast. The cytotrophoblast and the syncytiotrophoblast. The cytotrophoblast, cyto means cellular. All right, so layer of cuboidal cells, something like this. So we have got a nucleus. We're calling it a cytotrophoblast. Next comes the syncytiotrophoblast. All right, wherein multiple nuclei is called a syncytium. All right, syncytial trophoblast, in which the syncytium, that is cytoplasm with nucleus, but there are no cell boundaries, there are no cell bond, multiple cell will coalesce to form, will group together to form a syncytium. All right, we call it syncytial trophoblast. Then we'll discuss different types of chorionic villi, the placental villi. We'll discuss how these villi are helpful in the transport of various important products. What are the subdivisions of the villi? We'll talk about it. We have got primary villi, we have got secondary villi, and then we have got the tertiary villi. Primary, secondary and tertiary villi. We'll discuss all that. All right. Then we'll talk about the placenta, the normal weight of the placenta, types of the placenta, shapes of the placenta. We'll talk about it very quickly. Then we'll talk about how this together, the growth of the embryo and the placenta are contributing to the well-being of the fetus. All right. So let us begin our discussion with the formation of placenta. In this chapter, the first thing that we'll be discussing in detail is the formation of placenta. Formation of placenta. All right. So placenta is nothing but it is an organ which is contributed by both the fetus and the mother. So we call it feto-maternal organ. The placenta connects the growing embryo with the pregnant uterus, so that it's such an important organ that its normal physiology and its normal anatomy is actually responsible for the well-being of the fetus. And also many complications of the pregnancy arise because of the disease in the placenta. Lot of complications in pregnancy. So I'll discuss that when I'll be discussing obstetrics topic. Here please remember that placenta, the normal placenta is 500 grams in weight. Its normal weight is 500 grams. It has got two surfaces. It is a disc-like structure. It is a disc-like structure. Coin-like structure, discoid, slightly biconcave, biconvex, slightly biconvex. It has got two surfaces. So the surface towards the uterus, you are calling it as maternal surface of the placenta. Surface towards the fetus, you are calling it as a fetal surface of the placenta. Fetal surface of the placenta. The maternal surface of the placenta is irregular and it is composed of 15 to 20 small lobes. Small lobes. The fetal surface is smooth and it is covered by an important membrane called as amnion. The fetal surface of the placenta gives rise to attachment of the umbilical cord. And the umbilical cord is attached to the center of this fetal surface. The anatomy of umbilical cord, yesterday I told you. The anatomy of umbilical cord, which consists of two arteries and two veins first, but the left vein is left and the right vein is obliterated. So it has got two arteries and one vein in the end. Two umbilical arteries and one umbilical vein. Which umbilical vein is left? The left-sided umbilical vein is left. The right-sided is obliterated. So these are what yesterday we discussed. So the formation of the placenta involves, important aspect is the formation of the placenta involves first implantation, So what is the process of implantation? What happens during implantation? Let us try to understand. So just before implantation, ovulation occurs first. After ovulation, fertilization should occur. So all these definitions I've already discussed in my previous classes. Ovulation followed by fertilization. Fertilization is followed by cleavage. Cleavage is followed by formation of the morula. Morula is followed by formation of the blastocyst. So if you closely observe, these are the chain of events that are occurring one after the other. Chain of events that are occurring one after the other, which are very, very important, one after the other. We know that blastocyst has got inner cell mass and the outer cell mass, and the development of blastocyst into embryonic disc occurs. Now, what happens is during implantation, what happens is the first and foremost point that occurs during implantation in the uterus is the decidua, formation of decidua. You call it decidual reaction. You call it decidual reaction. And after this, what happens is adhesion to the uterine endometrium. Adhesion means attachment. So formation of the decidua, then adhesion, then followed by penetration of the embryonic structures into the uterine endometrium. After that, what happens is there is differentiation of this uterine endometrium into the trophoblast. That is the cytotrophoblast and the syncytiotrophoblast. So formation of cytotrophoblast and syncytiotrophoblast occurs. After this, what happens is there are small spaces that are formed, the lacunar spaces that are formed in the syncytiotrophoblast. Alright, small spaces that are formed and chorionic villi are formed. So this is what marks the important steps in implantation. This is what marks the important steps in implantation. Alright, we all know that first ovulation, fertilization, then all these happen, and I need not go into the details of the ovulation, fertilization, cleavage, morula, blastocyst. We already discussed all these points. If you remember my last class, in my class I told you I have shown you one diagram. I'll just help you out with that picture again. So this was the picture which I was discussing in my last class. Alright, if you can see here, first ovulation. Alright. So ovulation here, then fertilization. Alright. Then there is differentiation into of zygote into 2-cell stage, 4-cell stage, 8-cell stage, like that. All these are occurring. After this, I emphasize on one more important point, that is the implanted blastocyst. At the level of implant, at the point of attachment, at the point of formation of the blastocyst, what happens is blastocyst undergoes implantation, and here what happens is the uterine endometrium. The uterine endometrium, if this is the uterus, I'm drawing only one half of the uterus, and if this is the, so the uterine endometrium, in this area, the cells inside the endometrium undergo proliferation to form a very thick mass of cells. This you are calling it as a decidua. This you are calling it as a decidua. And in this decidua, we have the embryo, growing embryo, which is undergoing various changes. We call it penetration, erosion, then formation of the placenta, differentiation of that placenta into the syncytiotrophoblast and cytotrophoblast, formation of primary villus, secondary villus, and tertiary villus. So this is what we need to understand with the help of this diagram. So coming back to the formation, the stages of implantation. Coming to the stages of implantation, the most important stage here is the differentiation of the trophoblast into cytotrophoblast and syncytiotrophoblast. Differentiation of trophoblast occurs around 8th day of fertilization. You'll have to remember this point. 8th day of development or 8th day after fertilization, what happens is the blastocyst is partially embedded into the decidua, and the cells around the embryonic pole outside, they get differentiated into cytotrophoblast. Please remember, the cytotrophoblast cells are also called as Langhans cells, or the cytotrophoblast layer is also called as the Langhans cell layer. The inner layer of the innermost layer of the cells are usually cuboid or low columnar. So they are cuboid like this, or low columnar cells. With a distinct nucleus, they have a lot of mitotic figures. Their cytoplasm is full of mitotic figures which are ready to divide. Alright. Then another differentiation we all know is syncytiotrophoblast. So we've got cytotrophoblast and syncytiotrophoblast. What is syncytiotrophoblast? Syncytiotrophoblast, the outer layer of it has got multinucleate cells. Alright, it has got multinucleate cells. It is also called as plasmodial layer. Another name is plasmodial layer. And it is devoid of the mitotic figures. It is devoid of the mitotic figures because it is formed after the division of cytotrophoblast cells. The cytotrophoblast cells, under differentiation, it gives rise to the syncytiotrophoblast. So depending on the site of implantation, you can call it. So if this is the uterus, depending on the site of implantation, we have got central implantation, wherein the blastocyst is implanted in the center of the uterine cavity. Central implantation. Other type of implantation is eccentric implantation, somewhere like this. Eccentric means away from the center. And interstitial implantation sometimes which occurs. So if this is the uterine wall, so inside the wall, we call it interstitial implantation, resulting in interstitial pregnancy, which is very, very difficult to grow or develop into a normal fetus, and sometimes abortions occur. All right, fetal loss occurs. So let us talk about the decidua now. So this is the uterus. This is the inner layer of the uterus. All right, inner layer of the uterus, like this. Innermost layer of the uterus, you are calling it as a endometrium. So what happens is, the uterine embryometrium after implantation of the blastocyst, it is called as decidua. All right, it is called as decidua. So what are the changes that are occurring? We have seen that because of secretory changes, because of presence of progesterone, because of progesterone hormone that is released from the corpus luteum, and under the influence of human chorionic gonadotropin, Now this hormone is secreted by the placenta and the ovary and the placenta. And if it is placenta, by the syncytial trophoblast cells of the placenta. By the syncytial trophoblast cells of the placenta, this hormone is secreted, and on the 17th to 18th day of menstrual cycle, that is 5 days after the fertilization or timing of implantation, the uterine endometrium is highly modified into an edematous and vascular structure. You call it decidual reaction. These decidual cells or the transformed cells, which increase in the size and number, and they help in the growth of the embryo. They help in the implantation of embryo by providing glycogen, lipids, and various other nutrients. All right. So based on the part of attachment of the embryo, so suppose embryo is attached somewhere here. All right, with the help of villi like this. So the decidua actually overgrows it. All right, and you call it, based on the part, the part of the endometrium that surrounds the embryo like a capsule, this part which is surrounding the embryo like a capsule, you are calling it as a decidua capsularis. You are calling it as a decidua capsularis. The part of the decidua that lines the rest of the uterine cavity, this area. I'll draw it with another color. This area you are calling it as a decidua parietalis. Decidua capsularis and decidua parietalis. Alright. So this is all about the decidual reaction, and this is all about the differentiation of the decidua. We have got decidua basalis, the part of the decidua, which is which contributes to the attachment somewhere here. I'll draw it with blue color. So this area is decidua basalis. So like this, we have got three parts of the decidua: decidua basalis, decidua capsularis, and decidua parietalis. Each of this has got important function. Now let us talk. We have spoken about the trophoblast cells. We have spoken about the decidua. Now let us discuss how the chorionic villi are formed. Alright, how the chorionic villi are formed. To understand this, you need to understand that decidua is divided into three parts, three types. So we have got decidua parietalis, we have got decidua capsularis, and then we have got decidua basalis. Is it not? Similarly, the chorion, so that part of the placenta which is contributed by the fetus, is divided into chorion frondosum and chorion levae. Chorion levae. Chorion levae. All right. The chorion frondosum and the basalis, they both are responsible for formation of the chorionic villi. These chorionic villi are divided into primary, secondary, and tertiary villi. Especially the secondary villi, the tertiary villi, They are helping in anchoring. All right, and this is what you need to understand. There are other parts of anchoring villi like floating villas, the ramus cori, the truncus cori and all. We'll discuss that later on. So for now, please remember the differentiation of trophoblast into two layers, that is the syncytiotrophoblast and cytotrophoblast. The cytotrophoblast, which is contributing to the cuboidal cells, the syncytiotrophoblast, which have got syncytium like cells. The chorion and formation of the chorionic villi is very, very important, which step which helps in the formation of the vasculature of the placenta, right? So if you can see the stages of formation of chorionic villi. So first, initially what happens is formation of the primary villus. What is the primary villus? The primary villus consists of a central core of cytotrophoblast cells, which is covered by a layer of syncytiotrophoblast cells. So that is primary villus. So we have got a layer of cytotrophoblast cells, which is covered by the syncytiotrophoblast layer, all right. The secondary villi are responsible for three layered. They show three layers. They have got outer syncytiotrophoblast, an intermediate cytotrophoblast layer, and inner extra embryonic layer. So they have got outer syncytiotrophoblast, then the intermediate cytotrophoblast layer. Next comes what? Next comes the inner layer of extraembryonic mesoderm. All right. Now, next it gets differentiated into tertiary villus. So what is a tertiary villus? The tertiary villus is same as secondary villus, except that the fetal, it has got the fetal capillaries, fetal blood vessels in the mesoderm. So it is same secondary villus plus blood vessels. So secondary villi plus blood vessels form what is known as tertiary villus. So how is this villus formed? Let us try to understand the process of villus formation. The process of villus formation, initially what happens is there is appearance of lacunae and trabeculae in the syncytiotrophoblast layer. So this is the syncytiotrophoblast layer. In the syncytiotrophoblast layer, what happens is, so small lacunae and trabeculae are formed like this. All right, like this. Small divisions or lacunae and trabeculae are formed. These small cavities or lacunae appearing in the layer of syncytiotrophoblast. Gradually, all these lacunae gradually all these lacunae will increase in size. Slowly these lacunae will increase in size, and they come to lie radially, especially around the blastocyst. Now these lacunae are separated from one another by a partition in the syncytium. These partitions, if you see it in a circular fashion, it's something like this. So we have got a lacunae like this. All right, the rest of the area is the syncytium. So this area is the syncytium. So you can imagine how these lacunae are radially. I'll draw it with a black color now. So radial lacunae like this. Like this, they are orienting radially. Radial lacunae are formed. All right, and these lacunae are filled off these lacunae are separated from one another by this partition of the syncytium, and these are open to the blood. So blood gets filled inside these lacunae like this. Now blood gets accumulated in these lacunae. Now, so in this, how the primary chorionic villus is formed. Each trabeculus is initially made up of entirely by the syncytiotrophoblast. After that, what happens, and full of the lacunae which are full of maternal blood. After that, what happens is a layer of cytotrophoblast begins to multiply and grow into each of these trabeculus. All right, so somewhere here I'll draw it with a green color. So we are seeing the cross section like this. So we have got this cytotrophoblast cell layer. Actually cells are more cuboidal. I'm just drawing it very fast so that I don't waste my time and your time also. So a layer of cytotrophoblast is formed around this. So this is a syncytotrophoblast, and this layer is the cytotrophoblast. Cytotrophoblast layer. And the trabeculus thus becomes to have a central core of cytotrophoblast. It has got a center of cytotrophoblast which is covered by the layer of syncytotrophoblast, and it is full of lacunar spaces which are full of maternal blood. So these are lacunae, and inside the lacunae we have got maternal blood. This is the primary villus. This is the primary chorionic villus. Now, I told you what is a primary chorionic villus. A primary chorionic villus is cytotrophoblast layer which is covered by the syncytotrophoblast layer. I told you cytotrophoblast layer, it is covered by syncytotrophoblast layer. The syncytotrophoblast layer in turn has got lacunae full of maternal blood. This is the primary villus. What is the secondary villus? Syncytotrophoblast cell surrounded by cytotrophoblast cell. Intermediate layer is cytotrophoblast cell, and innermost layer is the inner extraembryonic mesoderm layer. So now let us talk about the formation of the secondary villus. Formation of the secondary villus. So here what's happening is, so here where is the, you may ask me, sir, where is the embryo here? Please remember, embryo is somewhere here. I'll draw it with a blue color. So the residua like this, and here we have got the embryo. In this embryo we have got the embryonic disc. if you remember, the ectoderm, endoderm, and the mesoderm, and it has got a amniotic cavity, and it has got a yolk sac like this. Alright, it has got a small amniotic cavity and the yolk sac. Alright, so now what happens is the formation of secondary, the extra embryonic mesoderm invades the center of each primary villus. The extra embryonic mesoderm invades the center of each primary villus, and this each primary villus now has a core of mesoderm, which is covered by the cytotrophoblast cell, which is again covered by the syncytiotrophoblast cell. Cytotrophoblast cells, which is again covered by the syncytiotrophoblast cells. So this blue color area here, this is the extra embryonic mesoderm which is invading. So the innermost layer is the extra embryonic mesoderm. Alright, this part. Alright, this part is the extra embryonic mesoderm. So in a primary villus, if the central core is invaded by the extra embryonic mesoderm, you are calling it as a secondary villus. Secondary villus has got three layers: centermost extra embryonic mesoderm, after that the green color cytotrophoblast layer, after that the pink color of the syncytiotrophoblast layer. Next comes the formation of tertiary villus, that is the secondary villus with blood vessels. Now where are these blood vessels? Soon the blood vessels can be seen in the extra embryonic mesoderm. Blood vessels are visible in the extra embryonic mesoderm. So now this takes a shape of something like this. So we have got the growing fetus into head and something like this, and we have got layers like this, layers of the growing fetus. All right, this is surrounded by cytotrophoblast layer. It gets differentiated like this. So differentiation like this. I am taking time to draw because you need to understand the concept here. So the green colored layer that we are drawing here is the cytotrophoblast, which in turn consists of syncytiotrophoblast, which in turn consists of lacunae. The small lacunae which had are now surrounded by the cytotrophoblast. So you can just if you can imagine this diagram, you can understand. I'm just drawing the partitions of the cytotrophoblast. The triangular structures which I have drawn in green color, these are the lacunae, which are again surrounded by the cytotrophoblast. So that forms the secondary villus, is it not? The innermost structure is the extra embryonic mesoderm. After that we have got cytotrophoblast. Then the next layer is syncytiotrophoblast. I'm just showing you where the blood vessels are attached so that we understand the formation of tertiary villus, because secondary villus, along with secondary villus, the blood vessels, presence of blood vessels makes it tertiary villus. All right. So this is all the, now this pink color is a syncytial trophoblast. I hope you are able to follow this. Inside this we have got lacunae, and inside that lacunae we have got the maternal blood. I will not complicate much of this diagram. This entire area is the secondary, the dotted blue color line is the extraembryonic mesoderm. The dotted area of blue color is the extraembryonic mesoderm. So I am discussing the formation of the tertiary villus. So what happens, blood vessels can be seen in the mesoderm forming core of each villus. So each villus here will have a blood vessel like this. So these are the blood vessels. This is what I wanted to explain. So blood vessels or capillaries will be like this. All right. So a secondary villus with a blood vessel, you are calling it as a tertiary villus. This is what is the formation of the tertiary villus. Hope you have understood this. Now in this villus, each villus if you talk about, there are subdivisions of these villus. Each villus if you talk about, I'll draw each villus in front of you. Now I'll draw each villus so that the villus that is formed first and attached to the fetal side of the extraembryonic membrane mesoderm and on the maternal side of the cytotrophoblastic shell, they are called as anchoring villus. Each anchoring villus consists of a stem. Each anchoring villus stem is divided into small branches. Branch you are calling it as the ramus. All right, ramus cori. I'll tell you how. So we have got, now we'll give structure to this what you have learned just now. So we have got a layer like this. Something like this, right? Then we have got a so we have got the fetal surface of the placenta and the maternal surface. So this is the maternal surface, the black color one. Maternal surface, which is divided into maternal cotyledons. Maternal cotyledons. All right. If you see here, what happens is each of these, the umbilical artery will have, the umbilicus will have two arteries and one vein. Two arteries and one vein. So we have got two arteries like this. I'm drawing arteries in blue color. Why, I'll tell you later. So normally arteries we draw it in red color. Then we have one vein like this, central vein coming like this. All right. So this vein divides into multiple branches like this. Got it? And with each vein we have got divisions of arteries like this. So this we are calling it as a truncus, the ramus and the ramuli. So this we are calling it as a truncus, the ramus and the ramuli. So this is contributed by the decidua. Yellow color is contributed by the decidua here. That is the maternal surface of the placenta. Got it? Maternal surface of the placenta. The maternal surface of the placenta contributed by the decidua. Then we have got the fetal part of the placenta. We call it chorion frondosum. I told you which has got attachment to the umbilical, the umbilical cord and the amnion, the amniotic membrane. The maternal surface of the placenta is rough, irregular. I told you it is separated by the septa or cotyledons. So septa, small septa which are growing there, and cotyledon into 15 to 20 cotyledons normally. All right, and if the placenta is viewed from the maternal surface, so you can easily see the grooves of the septa. You can easily see the grooves of the septa. The fetal surface of the placenta is smooth because of the amniotic membrane. It has got attachment to the umbilical cord. The fetal, the division of this chorionic plate, the blood vessel division of this chorionic plate is something like this. So we have got first the villus, the anchoring villus. Anchoring villus divided into the ramus, the stem villus, we call this. So this is the anchoring villus, and then these are the branches. You are calling it as ramus. And ramus will divide into ramuli or rami. All right, ramuli corae we call it. So we need not go into that much details. This much is important to understand. The placenta. Now we'll draw an ultrastructure of the placenta. I will try to understand how the placenta is contributing to the connection, that is the blood vessels connection with the fetus. We'll try to understand. So we have got fetal surface on one side and maternal surface on the one side. On the other side, we'll try to understand with the help of this diagram now. All right. Say suppose in this area we have got so say suppose this is the uterus, wall of the uterus. We have got, say suppose this is the decidua or the uterine endometrium. So in this you can understand the importance of the cytotrophoblast cells. This layer which I am drawing now is the cytotrophoblast cell layer. Today a lot of diagrams, so you can increase the speed of this video, playback speed, so that you don't waste much time. I am drawing it for you so that you understand it properly. We have got this layer, you can call it a cytotrophoblast layer. We all know the cytotrophoblast layer is surrounded by a layer of the syncytiotrophoblast layer on the outer side and also on the inner side. So we have got outer syncytiotrophoblast, the inner syncytiotrophoblast layer. All right, and then I'll label everything. Just give me a second. Then if you can see the maternal villus like this. We have got again the contribution of again cytotrophoblast layer. So this is the cytotrophoblast layer. In this we have got blood vessels like this. So these are the blood vessels. I'm drawing it for you. The only reason is I want you to just follow the importance of the division of right. All right, so this is all syncytial trophoblast, the pink color syncytial trophoblast. Surrounding this we have got a lacunae full of maternal blood like this. All right, so now let us start labeling it quickly. We have got the uterine surface like this, uterine myometrium. This is the uterine myometrium. This is the decidua. This is the outer syncytial trophoblast. This is the cytotrophoblast. This is the inner syncytial trophoblast, inner syncytial trophoblast. This is the villus with the blood vessels. All right. This part is the chorionic plate. Which consists again of cytotrophoblast, the syncytial trophoblast, the cytotrophoblast, the syncytial trophoblast, the blood vessels. All right, and the innermost structure that is surrounding the blood vessels like this. So this is the amnion. Hope you understand this point. This is the amnion, right? Now this is all the layers of the placenta, and there is one important layer here called as Niette's membrane. Niette's layer, which is the toughest layer, outermost toughest layer of the placenta. This Niette's layer is present in the outer syncytial trophoblast layer here. So this is the Niette's membrane. So hope you have followed the lecture. This is all about understanding of the layers of the placenta. All right. As I told you, the placenta is a discoid in structure. Sometimes there are variations in the shape and size of the placenta. So sometimes it may be fenestrated, sometimes it may be bilobed structure, irregular structure, triangular structure. All those we'll discuss in obstetrics and gynecology class. All right. The umbilical cord also, yesterday I have discussed in detail the different types of umbilical cord. You can just go through the notes, and if you have any doubt, you can just definitely talk to me. We'll now talk about the twinning, the fetal twinning. See, what happens is, if there is fertilization of a single, if the sperm, that is the male gametocyte, and the ovum, that is the female gamete, when they combine to form, they give rise to a zygote. All right, and the process you are calling it as a fertilization. Fertilization. All right, the process you are calling it as a fertilization. All right, fertilization of a single ovum. Fertilization of a single ovum by a single sperm. You are calling it as a, and formation of a single zygote, you are calling it as a monozygotic fertilization. At the same time, so imagine there is fertilization of two separate ova, a sperm plus a ova. Because of two ovulations, a sperm plus an ova, forming two zygotes, one zygote, another zygote. So this type of fertilization, you are calling it as a dizygotic fertilization. dizygotic fertilization. So there are two types of twins. So if this zygote divides into two zygotes, here you have got a twin pregnancy, you are calling it as a monozygotic twin pregnancy. Whereas the second case I told you, dizygotic means two separate acts of fertilization is occurring. Two separate fertilizations, two separate zygotes, you are calling it as a dizygotic. Alright. If it's a two separate zygote and two separate fertilizations, here what will happen is the fetus will be may have two different sex. So they may have two different sex. So one male, one female, something like that. Two different sexes of the fetus is possible. Next is they are unidentical twins. They are not identical because two different fertilization, two different chromosomes. Hence, genetically they are not similar. Genetically they are dissimilar. And the second important fact is they have got two chorions and two amnions. Two amnions and two chorions. If you compare the same with the monozygotic twin, they always have same sex. Why? Because sex is determined at the time of fertilization and division of this zygote has occurred after fertilization. Hence, the sex is always same here. They are very much identical to each other. They are very, very much identical to each other. Alright. They are genetically very much similar. Genetic similarity is there. Genetic similarity is there. So here the division of the chorion and amnion is something like this. They are diamniotic, monochorionic usually. Diamniotic, monochorionic usually. Diamniotic, monochorionic usually. What is this amnion and chorion? We'll discuss that later on when we discuss actual twin chapter in obstetrics and gynecology. We'll discuss that. But for now, for our basics of understanding of embryology, these 2 points are important. We have got two types of twins, monozygotic twin and dizygotic twin. Monozygotic twin act of single fertilization, dizygotic twin act of two fertilizations. The monozygotic twin always they have same sex, whereas dizygotic they may have different sexes. They may have, I'm not saying they always have, they may have different sexes. And monozygotic twin is usually identical. Monozygotic twin usually have genetic similarities, and monozygotic twins they have diamniotic and monochorionic type of placenta, whereas the dizygotic twins have got diamniotic, two different amnions and two different chorions. So diamniotic dichorionic type of placenta. So this completes the topic of twinning. This completes the chapter 6 of the placenta, the fetal membranes, and the twinning chapter. If you have any doubts, please get back to me. I'll be happy to help you. And happy reading. Bye.